Hi, my name is Karen McRae-Johnson. I'm the Invivagen Specialist at DermaLife Research. This video is to provide you with information about Mycoplasma contamination and PlasmoTest. Mycoplasma is the smallest and simplest self-replicating organism. Mycoplasma contamination cannot be detected visually or even by optical microscopy. Mycoplasma contamination is problematic for a multitude of reasons. Mycoplasma can drastically alter cultures and skew results, for example, by altering cell metabolism, growth, and gene expression. Mycoplasma has also been shown to affect signaling pathways, such as by activating toll-like receptor 2 of immune cells. Additionally, mycoplasma can seriously impact data reliability and reproducibility, and as scientists, these are attributes that we strive for when producing data. Mycoplasma contamination is also highly infectious for all eukaryotic cell cultures, and this can lead to loss of precious cell lines and loss of money and time. Invivagen have developed a cell-based assay to easily detect mycoplasma called PlasmoTest. This assay has a simple readout. For instance, you just need to add a small amount of your sample to the Mycoplasma reporter cells. If mycoplasma is present, you can visually see a color change from a pink color to a purpley blue. And this can also be quantified using a spectrophotometer. And the optical density is in direct proportion to the amount of contamination. If no mycoplasma is present, no color change is observed, and there'll be no significant difference in optical density. In addition to the simple readout of PlasmoTest, other benefits include that it detects all Mycoplasma species with a very high sensitivity. The assay can be performed in a 96-well plate, meaning it can be used as a convenient method of routinely testing multiple cell lines. In addition to checking cell cultures, PlasmoTest can also be used to test biological reagents for contaminations, such as media and FBS. After receiving your PlasmoTest kit, frozen stocks of the Mycoplasma reporter cells can be made. Therefore, you won't need to actually repurchase the PlasmoTest kit in the future. As I mentioned, PlasmoTest is provided as a kit. This means that it comes with the Mycoplasma reporter cells as well as the products required for the assay, including the positive and negative controls. The PlasmoTest kit comes with sufficient reagents for up to 250 sample tests. Invivagen have products to restock PlasmoTest reagents and controls, such as their PlasmoTest reagent kit, which includes enough controls and reagents to test a further 500 samples. Or you can just purchase the PlasmoTest controls. Invivagen also have a variety of products to detect, prevent, and eliminate microbial contamination, as shown here in the table. For more information on Invivagen products, please check out our website, or feel free to contact me for any quote requests or if you have any questions. Thank you.